In the fall of 2001, 22 people became infected by letters containing traces of anthrax that had been sent through the United States mail. 5 of the victims later died of anthrax poisoning. Since then, the United States, as well as numerous other countries, have begun strengthening their tactics and protocols designed to prevent such attacks, both domestically and internationally. The goals of these protocols are to eventually manage early recognition of current and emerging pest and disease threats, initiate threat responses, and improve emergency preparedness and response. Throughout the world, governments have focused on the technological advancements regarding weapons of mass destruction, such as nuclear weapons, that are more commonly associated with terrorist attacks. However, as a whole, the nation is nowhere close to being as prepared for bioterrorism as they should be. Bioterrorism can be defined as a form of terrorism involving the release of toxic biological agents. Many times, these biological pathogens have legitimate uses in research and industry, especially agricultural industries. But in the wrong hands, they could easily be misused by terrorists. It is unique in the way that it can be targeted at a larger population or a single person, making the detection and defense more complicated. Bioterrorism can possibly be prevented by the utilization of both biosecurity and biosafety. Many people use these two terms interchangeably, but for the sake of gaining a clear understanding of the future actions needed to defend against deadly pandemics, it is important to know the difference between them. Biosecurity is a strategic and integrated approach to analyzing and managing relevant risks to human, animal, and plant life, as well as the health and associated risks to the environment. This is a multifaceted process of interaction between humans and non-human biota, anthropogenic environmental and ecological factors, and socio-economic and political pressures. Biosecurity also tends to involve policies and procedures designed to prevent theft, diversion, or malicious use of dangerous pathogens and toxins. On the other hand, biosafety directly involves the technologies and procedures that use dangerous pathogens. This aims to prevent accidental infections of biomedical researchers, as well as the release of biohazards from research laboratories, whether it is directly or indirectly. Spread of pathogens can also occur when transporting specimens to or from the facilities and other locations. Usually, biosafety goes hand in hand with biocontainment. This is the process in which impermeable barriers or filters are placed between both the infectious agents and researchers, and between the laboratories and the outside world. Two other key terms that are frequently mentioned are biorisk and biohazard. Biorisk can be defined as the risk associated with biological substances and infectious agents. These substances and agents pose a risk to health, specifically human health, and are known as biohazards. Identification and classification of biorisks are often conducted in laboratory settings using biorisk assessments. The security parameters used with biosafety and biosecurity are used to control the unique characteristics that pathogens exhibit. In retrospect, it is much easier to detect, prevent, and defend against other types of terrorist attacks. Biological agents such as deadly pathogens are microscopic and rapidly developing, much faster than health professionals and researchers can account for. More often than not, dangerous biological agents exist naturally in the environment and can rapidly reproduce from even the smallest sample, as long as it is living within suitable environmental conditions. Many of these pathogens and viruses are able to be genetically engineered, turning a semi-harmful biological agent into a deadly specimen able to infect masses of people in a short period of time. Negotiating global standards that restrict access to dangerous pathogens would reduce the threat of bioterrorism, while also reinforcing the legal prohibitions on the development, production, and stockpiling of biological and toxic weapons. Many protocols suggested in the past have lacked a formal mechanism for monitoring and verifying the use of biohazards, transparency, compliance, and inspection procedures. With most of the dangerous pathogens being used in research laboratories, numerous inspections of facility security have been conducted. A United States Department of Agriculture inspector found that 124 of the labs in use were vulnerable to theft. Investigations in the U.S. Pentagon's biofacility and the U.S. Army Medical Research Institute of Infectious Diseases also found similar results regarding lack of lab security. This was not limited to susceptibility to theft, but also included failures to account for samples of pathogens due to poor internal controls and record keeping by staff. It is often insinuated within the media that breaches in biosecurity and biosafety are limited to members of terrorist groups, but the characterization of terrorists may include a broad range of groups. Yes, criminal or terrorist members may attempt theft, but visiting scientists, students, and short-term contractors may be provoked to engage in the theft of the dangerous yet expensive pathogens. It is also not unlikely for the theft to be conducted from the inside. Many times, trusted members of the scientific or technical staff become motivated to steal their own facility specimens. Some common motives are due to the workplace issues, financial insecurities, external threats such as from individuals unable to get into the facility themselves, and/or psychological problems. They are often trusted with unescorted access to the facility and become familiar with what is contained and where. Biosecurity is underrepresented as a policy and financial priority among countries and international institutions working to address global health security and to counter the risk associated with weapons of mass destruction. Many stakeholders do not invest in biosecurity assistance, and financial commitments are not well tracked. The stakeholders involved in global biosecurity can be divided into two groups: national and international. The national stakeholders are the sector-specific government agencies that have a primary interest in dealing with biosecurity threats, but industry, scientific research institutes, specialist interest groups, non-governmental organizations, and the general public also take play. International stakeholders are the international standard-setting organizations, international bodies, and international legal instruments and agreements that constitute the governance framework for biosecurity. A successful implementation of a harmonized and integrated approach requires a clear policy and legal framework, an institutional framework that defines the roles and responsibilities of relevant stakeholders, risk analysis, testing and control, and a system for communication and information exchange. Global biosecurity engagement programs are designed to prevent the harmful use of biological agents and pathogens. The goals of these programs have traditionally been quantitative in nature. For example, increasing the number of biological agents secured and number of scientists engaged. The United States and Algeria formed a partnership to develop a biorisk management program. Scientists and professionals within Algeria's public and animal health sectors were trained by health professionals from the U.S. to gain the skills necessary to deal with biological pathogens. This is an example of how important it is for larger, more developed countries to aid developing nations in order to fill the gap in which pathogens can be more easily spread. The primary goal of global biosecurity standards should be to strengthen the weakest links, those countries that research laboratories and culture collections are so poorly secured that terrorists could penetrate them easily. In 2014, more than 24 countries and organizations launched a global health security agenda to accelerate global capability to prevent, detect, and rapidly respond to biological threats. Many of them are also affiliated with the Global Partnership Against the Spread of Weapons and Materials of Mass Destruction. This organization internationally funds nations in order to improve biosecurity worldwide. 13 countries reportedly give a total of more than $470 million for capacity building programs. Approximately $370 million went to projects specifically targeted at improving biosecurity, biosafety, and countering bioterrorism. The projects were implemented by the United States, Canada, the European Union, Germany, the United Kingdom, Netherlands, Norway, Denmark, Finland, France, and Sweden. The United States was responsible for approximately $245 million of this funding. If countries do not take biosecurity into consideration more, future bioterrorism could result in long-term disasters causing regional instability and challenging international security. To prevent such misuse, biosecurity systems require an integrated approach, physical protection, access controls, materials accountability, and personal screening. The global guidelines should include, as a minimum, the registration and licensing of facilities that work with dangerous biological agents, based on an agreed list of pathogens and toxins that can be readily updated. Physical security, access controls at laboratories and storage within experiments, background checks on laboratory personnel, and an emergency plan for responding to breaches in security are also necessary. Also, procedures regarding conducting inventories and audit samples of collections, documenting the chain of custody of dangerous pathogens outside the access-controlled areas, and verifying the destruction of working stocks at the end of an experiment should be included. Lastly, training of scientists and technicians is appropriate laboratory practice is top priority to reduce spread of pathogens, as well as the establishment of regulations regarding the safe and secure transportation of hazardous goods, controls on imports and exports, and verification of the declared end use.